Cytologic description of human neutrophils. Human neutrophils, a type of white blood cell, have distinctive cytological characteristics. Nucleus. Neutrophils typically have a segmented or multilobed nucleus, which sets them apart from other white blood cells. The nucleus can appear as 2 to 5 lobes connected by thin strands of chromatin. Cytoplasm. Neutrophil cytoplasm is pale and granular in appearance. It contains specific granules that are rich in enzymes and proteins needed for their immune functions. Size. Neutrophils are among the smallest white blood cells, measuring around 10 to 12 micrometers in diameter. Granules. Neutrophils have two types of granules: primary, azurophilic, granules, which contain enzymes like myeloperoxidase, and secondary, specific, granules, which contain various proteins involved in microbial killing. Cytoplasmic vacuoles. In response to infections, neutrophils may develop cytoplasmic vacuoles to store ingested microorganisms. Neutral staining. Neutrophils typically stain neutrally, meaning they do not take up acidic or basic dyes as intensely as other blood cells. This property is often used for their identification in laboratory settings. Motility. Neutrophils are highly motile and can move rapidly towards sites of infection or inflammation. Their ability to chemotax, move in response to chemical signals, is a critical part of their function. These cytological characteristics make neutrophils easily recognizable under a microscope, and are essential for their roles in the body's immune response. Their segmented nucleus and granular cytoplasm are distinctive features used to identify and study these immune cells in laboratory settings. Origin of human neutrophils. The origin of human neutrophils lies in the process of hematopoiesis, which occurs primarily in the bone marrow. Here's a brief overview of the origin of human neutrophils. Hematopoietic stem cells, HSCs. Neutrophils, like all blood cells, originate from hematopoietic stem cells. These stem cells are located in the bone marrow and have the remarkable ability to differentiate into various blood cell types. Myeloid progenitor cells. Hematopoietic stem cells differentiate into myeloid progenitor cells, which are committed to giving rise to cells of the myeloid lineage, including neutrophils. Neutrophil precursors. The myeloid progenitor cells further differentiate into specific precursor cells for neutrophils. These precursor cells go through several stages of development, including myeloblasts, promyelocytes, myelocytes, metamyelocytes, band cells, and eventually mature neutrophils. Maturation. Throughout these stages, the cells undergo morphological changes and accumulate granules containing enzymes and proteins that are essential for their immune functions. Release into circulation. Once the neutrophils reach their mature, segmented form, they are released from the bone marrow into the bloodstream. From the bloodstream, neutrophils can be quickly recruited to sites of infection or inflammation, where they play a pivotal role in the immune response by phagocytosing, engulfing, and destroying invading pathogens. Understanding the origin of neutrophils is fundamental to appreciating their importance in the immune system. Birth of human neutrophils. The birth of human neutrophils, also known as neutropoiesis, is a fascinating process within the bone marrow. Neutrophils are a type of white blood cell and play a crucial role in the immune system's defense against infections. During their development, they go through several stages. Hematopoietic stem cells. Neutrophils originate from hematopoietic stem cells in the bone marrow. These stem cells have the potential to develop into various blood cell types, including neutrophils. Myeloblasts. Hematopoietic stem cells differentiate into myeloblasts. These are early-stage neutrophils that undergo a series of transformations as they mature. Promyelocytes. Myeloblasts progress into promyelocytes, which develop specific granules containing enzymes and proteins needed for their functions. Myelocytes. The next stage is myelocytes, where the cells begin to take on the characteristic appearance of mature neutrophils. Metamyelocytes. Metamyelocytes have a distinctive kidney-shaped nucleus, one of the defining features of neutrophils. Band cells. These cells have a U-shaped nucleus and are considered slightly more mature. Mature neutrophils. Finally, band cells develop into fully mature neutrophils with a segmented, multilobed nucleus. At this stage, they are released into the bloodstream. Mature neutrophils are then circulated throughout the body and are capable of recognizing and phagocytosing, engulfing, foreign invaders such as bacteria, fungi, and other pathogens. Understanding the birth and development of neutrophils is crucial to appreciating their vital role in the immune system. Functions of human neutrophils. Human neutrophils are a critical component of the immune system and serve several essential functions. Phagocytosis. Neutrophils are expert phagocytes, meaning they can engulf and digest foreign particles, such as bacteria, fungi, and other pathogens. This process helps eliminate the invaders from the body. Chemotaxis. Neutrophils can sense chemical signals released by damaged tissues or invading microorganisms. They follow these signals to migrate to the site of infection or inflammation. Inflammation response. Neutrophils play a key role in the early stages of the inflammatory response. They release various chemicals and enzymes to recruit other immune cells to the site of infection or injury. Microbial killing. Neutrophils contain granules filled with enzymes and antimicrobial proteins. They release these substances to destroy engulfed pathogens. Respiratory burst. Neutrophils can undergo a respiratory burst, generating reactive oxygen species, ROS, to help kill bacteria and fungi. Extracellular traps. Nets. Neutrophils can release DNA and proteins to form web-like structures called neutrophil extracellular traps, nets. These traps immobilize and kill microbes. Tissue repair. Neutrophils also participate in the cleanup phase of inflammation by removing cellular debris and apoptotic neutrophils, promoting tissue repair. Immune regulation. Neutrophils can influence the immune response by interacting with other immune cells and modulating the overall immune reaction. Overall, neutrophils are crucial for the body's defense against infections and are often the first responders to sites of infection and inflammation. They help maintain the balance between protecting the body from harmful invaders and preventing excessive inflammation.